please bear with me. And it's an honor to be here with each one of you. I'm calling, or at least zooming in from Malaysia, and I'm excited today for this opportunity to share with each one of you a little bit about the work that we do. So without further ado, let me just get started with things. Okay, so my name is Josiah, and I work basically with Lab-Ind Resources here in Malaysia. I have been involved in the company for the last 4 years, and my role is basically in business development, operations programming, and a little bit of bioinformatics. All right. I will explain a little bit more about Lab-Ind Resources as we go along. For today's presentation, I'm taking it as an opportunity for us to learn from one another. It's not just me, hopefully, speaking at you, but I would love to engage you in some of the questions, your comments in the chat and the Q&A later as well. And this is also an opportunity, I think, for reimagination. And what we're going to talk about today is a little bit of molecular diagnostics. And for many of you involved in the animal health industry, I think it's an opportunity to reimagine how molecular diagnostics can be done within your sector. Okay, so again, I work with Lab-Ind Resources, and we are a Malaysian-based lab company. And our specialization basically is in the development and application of molecular diagnostics and the complementary technologies around it. Our sectoral expertise is in animal health, food safety, and other underserved industrial challenges, typically in environment, soil. These are things that are services that are not typically very common, and we are working to provide molecular solutions to that industrial challenge, right? Just a little bit more background about us. As an organization, what we do is we have our own lab service, and our lab is accredited 17025 for quality management system here in Malaysia. Our team is involved not just in Malaysia, but also regionally in Southeast Asia, and as far as the Middle East, and a little bit of work in Africa, which I'll touch on a little bit later. Okay, so here are some of our partners, international collaborators, government officials that we work with. In addition to that, we also package the kind of services that we provide in our lab to other labs in the region. I'm sure you all know labs are notoriously geographically restricted. Right, we cannot move a lab from one country to another very easily, and so there are labs all over the world. And what we're doing is packaging some of the services that we have in our own lab for them as products, basically. And then also we're involved quite a bit in knowledge transfer, so capacity and capability building. Here's a picture of our team, Mr. Leong, who will be speaking in the next presentation, doing work in Bangladesh. And then some of the work that we do closer to home with students and with teachers, high school teachers and students. All right. So I hope that gives you a bit of a sense of what we do. Here are some of our international partners, service providers that we work with in Southeast Asia, and also the competent government authorities. As I mentioned, we are also doing work, a little bit of work in the African continent with WorldFish. And so WorldFish has their Give Tilapia program, and we are involved with them to provide them with fish health assessments for Give Tilapia in Nigeria, but also in some other countries on the African continent. So we do have that little connection, right? So that's a bit of a background and introduction. Today, actually, for this first presentation, I wanted to set the scene for us. I think what is amazing about this presentation and this opportunity with the EVMEDS team is that this is not a singular presentation, and it does not live in isolation, but there will be multiple presentations after this that will build upon the work that we're doing here today, and then the next week and the following week as well. So today we're just going to start with the foundational basics, which is my role, to speak to you a little bit about molecular diagnostics. But I hope that as you read kind of through this timeline, you will get a sense that we are moving from this abstract idea of molecular diagnostics to a more concrete and practical application. So my colleagues next week will talk about how exactly molecular diagnostics is applied specifically to the aquaculture and poultry sector. And then following that, Mr. Robin, who is our director, one of the co-founders of the company, will be talking about how molecular diagnostics can be started in the country. So in Malaysia, when we first started about 12 years ago, maybe 12 to 15 years ago, molecular diagnostics was still something that was unknown, right? And there were a lot of challenges to get started, but then we've navigated through that. And that model has been applied not just in Malaysia, but in Myanmar and Bangladesh for impactful, but most importantly, for sustainable use of molecular diagnostics in our work. All right. And so I hope you see that move from abstraction to concrete reality, and that you follow us through over the next 3 weeks. Right. So without further ado, let's jump straight into it. I'm launching a poll basically for you, and on the bottom right of the screen, I will have at certain points this poll 1 thing. So if you don't mind, if you can just answer the first question. There are 5 questions, but we'll go through it sequentially. I think that will give us a little bit of an idea of where everyone is from and what you all are doing. Okay, so poll 1. All right, I'll jump into it. So let's start with the what. Today is a primer to molecular diagnostics. Well, let's strip that all down and talk about what exactly is a diagnostic. I think that is very important to begin with, right? And so combining the definitions from the Global Animal Health Association and also Roche, diagnostics basically are technologies that detect pathogens, diseases, or conditions, and then form the basis upon which clinical decisions are made, all right? And so these are the technologies that you and I use to discover hidden diseases or pathogens, and then based on your discovery, you're going to make certain plans, right? So that's what a diagnostic is. Now, that idea of diagnostic as technology will sit within specific paradigms, and the paradigms that we're looking at are going to be diagnosis versus prevention, right? So diagnosis is basically the act of identifying the disease through the signs and the symptoms that are manifest, while prevention is basically the idea of trying to stop that manifestation from happening, right? And so diagnostics is going to sit within one of those 2 paradigms, sometimes both. And I think the Global Animal Health Association speaks of diagnostics as identifying health issues before they are otherwise able to be detected via gross clinical signs. And they add that early detection reduces the duration and intensity of treatment required, while also protecting other livestock. And so for those of you who are in the veterinary profession, especially working on commercial farms and animals, this whole idea of protecting other livestock is key when we're talking about prevention, because you're not just dealing with a single individual animal, but you're also typically dealing with a flock, a community, right? And so that whole idea of diagnostics within prevention is gaining a lot of traction. Okay, in their report, again, the Global Animal Health Association, they talk of diagnostics, the future of diagnostics, as to track an animal's health trends over time and detect hidden issues before they worsen. Recognition of diagnostics as a key tool for prevention, not simply diagnosis of an acute issue, will drive better overall health for animals. And so we all know this axiom that prevention is better than cure. And that is kind of the idea that we would like to operate within when we talk about molecular diagnostics today. All right, so we're going to talk about molecular diagnostics within the paradigm of prevention, and not necessarily just diagnosis. Okay, this is important as the world moves into this whole One Health concept, where environmental health, animal health, and human health are interlinked. That prevention becomes an important part of our methodology, right? Because once you prevent, without prevention, once something goes wrong in one, it's going to affect the other sectors as well, right? So just to kind of encapsulate all of that, right? Diagnosis is basically the application to a sick individual whose gross clinical signs and symptoms can be seen. With diagnosis, you typically are confirming the disease that's present and what is it. And then it's all about corrective action. So it has happened, and now you have to deal with it to prevent it from happening again, right? But when we talk about the other paradigm of prevention, we're talking about a physically healthy population, and we want to find potentially disease that may be happening within the population, but like is not seen yet. And this whole idea is for prevention, right? Before the disease manifests and spread, you want to catch and stop it from spreading to that wider community. And so this is the direction, the paradigm that we want to operate within, or that we believe you should begin to think about operating within. Okay, so today again we are talking about molecular diagnostics, but I'd like to also give a quick run through of some of the other diagnostic methodologies that are available, right? There are biological methodologies and also chemical methodologies, of course, but I won't focus so much on the chemical side because that is not my area of expertise. So today we'll look at diagnostic methods of the biological side, right? And there are four main ones that I would clump things together. Would be cytology and histology, microbiology, serology, and then of course molecular diagnostics right at the end. Okay, so just to prime you all to think about these again, for those of you who are unaware, cytology and histology basically provide us with a cell level view of what's happening. Cytology specifically for cells and histology for tissues and their structures. This photo is I took from one of our participants, actually, Dr. Kunda from Zambia. And this is a picture that of a histological sample that he's done. Yeah, quite beautiful. But without any context, it is very difficult for you to tell what exactly is going on. I myself can't really tell what's going on here. And so this methodology, this technology, requires an expert eye and also very skillful hands in preparing this sampling. All right. Basically, what the purpose of cytology and histology is to assess the cellular and tissue response to the presence of a pathogenic agent. And usually that happens when the gross clinical signs are already manifested, right? And so when we're talking about the paradigm of prevention, this does not, as a methodology, work very well because you need to go through, you're already in that disease stage, right? Before you can do cytology or histology, okay? So this is one diagnostic method. I believe, let me see. Yes, so we are now at poll 3. I'm not sure if you guys are answering your poll questions, but yeah, if you could drop your poll questions, that would be great. Your poll answers, sorry. Okay. The second diagnostic method that we just want to briefly cover is in microbiology, right? So microbiology basically provides a cell level view again, but also a biochemical and functional view. And this is done basically through the culturing and cultivation of the pathogenic agent and then subjecting that cultivated pathogenic agent to different types of assays that can measure I think, responses, right? And that gives you an idea of the functionality of that cell. And through that functionality, you can begin to piece together a type of identity. However, some of the limitations of microbiology is that typically there's a list of pathogens that we know about, but I'm sure that there's a far wider list of pathogens that we do not know about, right? And so that is the challenge with microbiology. Not all organisms also are culturable. Typically, when we culture things, it will be aerobic microbes, right? But then there are whole classes that are not easily cultivated within a lab, okay? Again, microbiology as a diagnostic method is scalable, but up to a point. If you're talking about, hundreds of samples, you'll be basically having a tower of Petri dishes, okay? And then one of the limitations still is that it is a time-consuming process. Microbiology will require between, anywhere between 48 to maybe 120 hours, right, of work to get done. In terms of cultivation, then in terms of subjecting it to different assays. However, if you can cultivate your microbe of choice, then that is still the industrial gold standard by which determinations about the identity are made, right? So if you can culture E. coli, you can confidently say this is E. coli or Salmonella or Listeria or whatever, right? So that's microbiology as a diagnostic method. Okay, and then serology. Serology is basically looking at blood and other fluids, saliva. I think now in this post-pandemic era that we live in, it is quite a ubiquitous technology. You probably all have seen it if you are traveling internationally or even locally. Rapid test kits for your nose and also for saliva are freely available, at least in Southeast Asia. And serology immunoassays basically fall within the same category of products. And one, serology looks for antibodies, which are defense proteins that are generated by the immune system in response, right, to a pathogen. And then immunoassays will look for the antigens, which are basically the antigenic, the proteins of the pathogen, right? So two sides of the same coin. As a technology, serology is incredibly easy and low cost and fast. And I'm sure if you've used that, you'd experience it. However, one of the challenges, again, is that antibodies are produced after exposure to a pathogen, right? So that is one of the difficulty there is that within the prevention paradigm, and we're talking about a prevention paradigm here, after exposure is a little too late, right? And so we are looking for a method or a tool that will enable us to detect very, very shortly on early exposure. Sorry. And just to clarify, but when I say after exposure, I mean a few days, maybe a week plus, before the antibody levels are produced. Okay, so that brings us to molecular diagnostics. For those of you who are unfamiliar, molecular diagnostics basically is diagnostics on a genetic level. All right, so we're talking about nucleic acid, DNA, or RNA. And I've abbreviated molecular diagnostics as MDX, okay? And basically, with molecular diagnostics, we're able to detect an incredibly low amount of pathogen, and I'll explain to you how that is done through the use of that nucleic acid, okay? And because we are able to detect an incredibly low amount of pathogens, that enables us to work very well within this preventive paradigm that we are looking for, which is to catch infections very, very early, right after exposure, and then which then translates into a reduction in treatment intensity and also time. Okay, molecular diagnostics is incredibly scalable. 96 samples within 2 hours is a norm, and you can you can do way more than that over the course of the 8-hour day. And so that's one of its strengths. However, the challenges are that infrastructure costs are high, and there is every technology has its limitations, and I think this is one of its main limitations. Okay. Now, just diving a little bit deeper into this whole idea of molecular diagnostics, which is why we're all here today. Basically, when I talk about molecular diagnostics, I'm talking about nucleic acids specifically, all right? And nucleic acids basically work on the genetic level. The technology, molecular diagnostics, is based upon typically PCR, or the polymerase chain reaction technology. And the principle behind that is basically the replication of a unique target genetic sequence in order to produce an interpretable and intense signal. So if you look at this schematic here on the right, basically every living thing, pathogen or otherwise, has within it a genetic code, right? DNA or RNA, some sort of nucleic acid. With PCR, once we extract that DNA from that pathogen, we artificially boost the amount of a unique DNA sequence that belongs only to the pathogen. And by artificially boosting that, we are able to tell the absence or presence of that pathogen, right? And so that replication, that artificial replication, increases the signal intensity of that pathogen's nucleic acid profile, okay? Now, so if you think about that, because we're artificially replicating that nucleic acid profile, the starting material, the amount of pathogen required for us to get a signal is actually very low, right? Because if we can artificially boost that signal, then it doesn't really require a huge amount of starting material. And so this actually enables us to catch infections in early exposure, right? So when typically you're exposed to a virus, the amount of virus usually is a little quite small at the start, but then over time it starts replicating within your body, right? So once the viral load gets too high, that's when you start seeing those gross clinical symptoms. We're able to catch it very early on when that amount of virus or pathogen is still very low. Okay, so that's the strength basically of molecular diagnostics. So here's a quick comparative kind of table for you with MDX, and these are kind of 3 MDX technologies, all based off the polymerase chain reaction technology. You may see that for serology and the immunoassay, or basically the RTK that we do use, right? The sensitivity is from 10,000 to about 1 million virus copies, right? And in terms of specificity, we can classify that as being pretty low. But with PCR or point of care type PCR, LAMP-based PCR or real-time PCR, you can see that the amount of virus required is incredibly low. And so that allows us, in terms of our objectives, to do screening or prevention, diagnostics, rather than just the diagnosis when the clinical symptoms are already present, okay? So within the kind of workflow, the veterinary workflow for medicine, here you can think about it, a rapid molecular test with the results, and you can test against a battery of different pathogens. You're not just testing for one, maybe you could test for 4, or 5, or 6, 7, or 8, right? At one time, you could come up with a very targeted treatment because you know what is there, and then achieve optimal outcomes within the shortest amount of time. Whereas in the case of, say, microbiology, you typically there is already gross clinical signs that are there, and then you have to, based on what you can gather, the information you can gather, start a treatment. But then if the treatment doesn't work, right, then you have this whole secondary line that you have to do, culturing, antibiotic tests, and that basically results in delayed treatments and then delayed patient outcomes, right? Now, this is not to say that microbiology is terrible or anything, but this is to frame it within the veterinary context. And if you're working from as a commercial veterinarian, this is something that you'll have to think about and consider, right? Because time is basically of the essence, okay? So brief summary for all of us here. Some of the advantages of MDX is basically that it's applicable across many organisms. As I mentioned, all living things, and then virus, if you count it as a living thing, you will have a DNA profile of some sort. And so the moment you know that it has a DNA profile, you can begin to design assays and tests to identify that organism, okay? So the tests for bacteria and virus and fungi, all of these things, it does not change very much in terms of the workflow. In terms of scale and throughput, it scales very well, and you don't require much to do a lot. Turnaround time is pretty fast. PCR technologies take between 2 to maybe 4 hours, depending on how skilled your technician is, and it can be applied across the value chain. So for us in Lab-Ind Resources, we don't just test from the farm, for example, but we're also testing with when the farm goes to the processors to be processed, and then from processing before it's exported, you could use it all across this whole value chain, right? Some of the disadvantages of MDX would be some of the technical complexities here. I didn't have time to talk about the full mechanism, but I think there are plenty of videos talking about PCR and the requirements, and it does require skilled technicians to operate. But once you have a workflow that's fixed, then it becomes much easier, right? And then the cost, infrastructure and the materials for the testing. I do believe that this is changing as technology improves. The costs are going down and the tools are becoming cheaper and easier to be used. And so this is kind of, we're in that wave right now. All right. Now I wanted to talk a little bit more, building upon that molecular thing, to touch upon just this new technology of next generation sequencing. It's not new, it's on the rise now. I think it's the next hot thing, right? And with next generation sequencing, some of the applications are basically a look at the genome level view of an organism. If you recall with PCR that I was talking before about, you're only looking at a specific sequence, a short sequence, maybe this area of an organism. But with a genome level view, you're able to get the full profile of that organism. And you can tell using that profile further functional characteristics, right? Does that organism have antimicrobial resistance? Where is this organism from? So these are some of the information that can be retrieved with sequencing technology, right? You can also use sequencing technology to get a compositional view. And so each line, each bar here represents a single sample from a gill of a fish that we for some work that we've done, right? And we typically talk about bacteria almost as though they live in isolation, but I think reality is that bacteria live in communities, right? They're communities of bacteria operating together, right? And so in each bar, these different colors each represent a unique bacterial species. And so this information, both the genome level and or the composition of a bacterial community, enables us as veterinarians, as researchers, laboratory technologists to make better decisions. There are companies that are already looking at this whole idea of the microbiome, the microbial community, and how different proportions of bacteria result in different effects, right, for the organism. Not in aquaculture, but also in poultry with respect to gut health. These are some of the new technologies that are now being rolled out. But I think the strength of next generation sequencing is also its is a double-edged sword. It's also its weakness. There's a lot of information and work that is required to navigate from just the raw information into something that is useful like this or like this, right? And then the process, there's a complexity process involved with next generation sequencing as well. Okay. So that was an incredibly quick run through quite a lot of different diagnostic technologies. I wanted to basically close it here today with this idea, this table, just to give you a brief kind of comparison, right? So we talked about cytology and histology today, microbiology, serology and immunoassays, PCR and next generation sequencing. Here are some of the complexities and how we grade it, with next generation sequencing being an incredibly complex kind of technology, and some of the costs associated with it, and also the objective, how we are applying these technologies, right? So within the paradigm of diagnosis, I think cytology, histology, microbiology, serology, and immunoassays are fantastic for diagnosis once you already start seeing the gross clinical symptoms. However, if you're talking about prevention, I think MDX would be a useful technology for prevention work. And I think the industry here in Southeast Asia at least, and I think across the world as well, will be moving in that direction towards prevention of diseases. All right. In summary, diagnostics technologies for detection of a pathogen, disease, or condition. There are many types of technologies and tools which we covered very briefly today, but which you can also look up. And I think I want to emphasize that each of it has a role to play, and each technology falls within its own unique paradigm. And I think that is important. I think that's necessary. When we're talking about, I think we're talking about animal health, this paradigm is now shifting from diagnosis to prevention, especially when we're talking about commercial animals, yeah? Because I think in a commercial context, early detection basically is the key to go with respect to ensuring animal health on a farm within the community. Okay, molecular diagnostics is what we covered today. It is a highly sensitive and highly specific technology. I think all you need to take away about molecular diagnostics is that where with anything that has genetic material, you could probably identify it, and you could identify it at incredibly small amounts, because the point of molecular diagnostics basically is to artificially boost the signal of that specific genetic material, all right? And so it is great for the prevention paradigm that we were talking about with respect to animal health. Some of the challenges are infrastructure, the tools, and also the technical capacities. It is still relatively high in terms of these costs, but as I mentioned before, this is changing. With new technologies, new innovations, there's greater opportunities now for people to do molecular diagnostics. I can speak to that personally. Here in Malaysia, we are already doing some molecular biology with high school students, right? Because the tools now are miniaturized and smaller, that we can bring it out to high schools and teach 17, 18-year-olds how to run PCR experiments on their bench and at their workspace. So that is pretty cool. Okay. Yeah. So thank you so much for 25, 30 minutes of your attention. I just wanted to come back to this today, just to remind you all that this introductory session does not live in isolation. Today is a primer to molecular diagnostics, a very brief, intense kind of primer to molecular diagnostics. But for those of you who are actually involved in industry, who are involved in research work, the next two sessions with my colleagues will be actual practical case studies of how molecular diagnostics can be used and applied in a commercial setting in the aquaculture and poultry sectors. Now, these are going to be examples from our own experiences here in Southeast Asia, but I'm sure that the principles that my colleagues will talk about can be applied and reimagined for a local application in Nigeria or the sub-region as well. Yeah, and then Mr. Robin, who's again our co-founders of director, has been bringing up molecular diagnostics within the Southeast Asian region as well, and so he will talk about how exactly do we get started with molecular diagnostics in a way that is impactful and also sustainable, because that sustainability is going to be key to actual usefulness of the technology, right? We don't just want to start something that is just for the fun of it, but we want it to last and to actually be impactful and to make a difference in our respective industries. So today, pretty abstract, but as we build, it will get more and more practical, and hopefully in 2023, my team at Lab-Ind Resources, along with the EVMEDS team, will be able to move things maybe into the sub-region, who knows? So we're building towards that direction, all right? So very briefly, here are some of our contact details. We also have your emails. If you're worried that we don't, you can contact us and we'll be happy to share with you the presentation slides as well. So thank you again, everybody. If you have any questions, the Q&A is always available. There's a Q&A tab over there. And then also, or you can type it in the chat and we'll be happy to go from there, right? So I will stop my sharing now and I'll pass the time back to Dr. Adebayo. And I look forward to interacting with each and every one of you. Thank you.
Yeah, thank you very much, Josiah. That was a great presentation, and I guess you've already been receiving some accolades in the chat box for that wonderful presentation. Yeah, thank you very much. For those people that were not able to see the presentation, sorry, I guess that should be the bad network. Sorry about that. But like we mentioned, Josiah also mentioned, you'll still be able to have access to the recording. So that you will be able to at least go through and get an idea of what we've done so far, because we're trying to ensure that we move from something like abstract to make it more practical each day as we continue with this webinar period. We should also note that we have a great plan, and we are not planning to start and leave you high and dry. No. So we have a great plan to ensure that you are not only stimulated, like Josiah mentioned, now we have secondary school even having access to PCR, and that should be very awesome. If secondary school students are having access to PCR, and then you're a graduate and you don't have access to PCR, then that's very strange. And we don't want it to be strange, really strange. And that is the reason why we're moving, like he mentioned, from something abstract to something more practical. And we're going to build that over the period of time. We also have great plan in 2023. So everything is going to eventually shape up with time. And we're going to continue this session also by next week. This same time next week, we're going to be meeting for more practical session with some other facilitators. So in case you have questions to ask, I know some people have already started typing in the question and answer box. So if you've not dropped your own, you can actually drop your own. Just click on the Q&A, that Q&A, click on it and type your question there so that you can have access to answer. We should also tell you that there is going to be a certificate award after the completion of this program, so you can actually tell other people that missed this section that they should come and learn something novel so that they can also be awarded a certificate for participating in this program. I'm going to go back to you now for a while, just Josiah, because they didn't see the poll. I don't know why, but yeah. So if there is anything you can do to help, that would be great. While other people are allowed to ask questions that may be that they may have in mind. So, can you do anything about the poll, or we should just leave that for next time?
Yeah, I think Dr. Adebayo, we can leave it for next time. I will go back and
Great.
figure out what is happening.
All right, no problem.
It's okay, everybody. Thank you.
So sorry about that.
Yes.
So you get to see the poll next time. Okay, yeah. So back to you, Josiah. We have question already, which you may like to attend to.
Yeah, sure. Okay, so this is from Okoli Solomon Chigluka. Hopefully, I'm pronouncing your name correctly, and if I'm not, I apologize. How easy can we get ELISA test kits and antigens in Nigeria from your company? So thank you for the question. Just to let you guys know, we as a company, we don't actually do ELISA testing. We don't produce our own kits for ELISA. Our focus is with PCR-based technologies, so molecular diagnostics, basically, the things that I was talking about today. However, if you're looking for serology and immunoassays, I think that technology is much more ubiquitous, and so any of the life sciences companies that you're looking at, or you could get in contact with Dr. Adebayo, and I'm sure he knows some of these organizations as well. So I hope that answers your questions. We don't do ELISA testing kits, but if you are interested in PCR-based testing kits, that is something that as an organization we do in Malaysia, and then also regionally, and we're moving globally as well. Thank you.
Okay, so let me quickly add to that. Yeah, so there is a difference between serology and molecular. So the concentration, or let me say the focus, is molecular based. But if you're interested in serology, I guess that's not going to be a problem. There are some organizations in the country that actually have access to supply ELISA kits. So you can contact me through the email, and I can also send that to the chat box now, so that you can easily contact me, and then we can get it across to you. So that's not going to be a problem. That's the reason why we are here in the country anyway. So for ELISA serologic test, yes, we can easily get across to me, and we can go ahead with that. So back to you, Josiah.
Okay, great. Yarginama, hopefully I'm pronouncing that correctly. Yarginama, thank you for your question. So the question is, are there any health issue concerns with like genome sequence on animals that could lead to mutations in humans? That's a great question. I just want to maybe reframe it a little bit, right? When we are talking about health concerns, typically what the conversation is about is genome modification, right? So genome modification is the act of modifying, of changing the genetic material within an organism. So most easily this could be, I think, thought of in probably plants. So let's say cotton, for example. Cotton that is resistant to certain insects, right? Because they produce a toxin. So that is a genomically modified organism. When we talk about genome sequencing, what we are talking about actually is not changing anything. What we are just reading the genetic material. Okay, so the thing that I was talking about with genome sequencing, oops, I think my camera died for a little bit. Sorry. When I was talking about genome sequencing, that is actually the act of just reading the DNA. Okay, so I hope that answers the question. So genome sequencing does not lead to mutations in humans. Genome modification If you are modifying a human, then yes, it will lead to some sort of mutation in a human. But animals, so if you modify an organism or an animal, right, and then the human eats that genomically modified animal, there is no evidence that there is a transfer of that modification to a human. So I hope that kind of makes sense. Yeah, right. Thank you.
Okay, yeah, Mr. Long also is trying to answer the question, but let me just come from the medical perspective
Yeah.
so that yeah, I may be able to also, well, Josiah has already said it. If it's just the sequence, if there is not any form of modification and consumption by human, then yeah, there is nothing that we should be afraid of. If animal is affected, it does not necessarily mean that a human being will experience any form of mutation. But you know, mutation, or let me say mutation in animal may also be caused by some viral particles. So this is where it may be a concern to human because if the virus that causes mutation in animals finds the way of jumping into human for whatever reason, with whatever means of transmission, yeah, there is also likelihood that if the virus finds a very good environment in human, yes, it can also cause mutation in human. So it depends on the cause of the change in the genomic sequence of the animal. That is what's going to inform, okay, if it's going to have any effect in human when they have interaction or when there is a consumption. And it even depends on what they use to process before consumption, because if you are aware, take for example, we have heat that can affect the structure of the virus, and because it can affect the structure of the virus, then it is not impossible. We may have that not even affecting human if the animal is boiled or put inside an environment where there was a very high temperature before being consumed. It could have deactivated the virus, and that may not have any effect eventually on human. So the question may need to be kind of rephrased, actually, to capture the thoughts in your mind in order to be able to provide the most adequate answer for that question. I hope that helps in any way. Okay, yeah. Looking forward to having you join up for another question. Thank you, Josiah, for saying that. So nice presentation and everything. I just want to be sure if there are no questions in the chat box before we close for today. Okay. Yeah, there is another question. Okay. Yeah, Josiah, you want to take that?
Hi, Okoli. Yes, thank you for your question. The question is, does your laboratory receive samples from other countries or independent researchers for sequencing? So my colleague, Mr. Leong, will give you a written answer. I can briefly say that we do sequencing projects, typically in our region, in Malaysia and also in Southeast Asia. I think when you're talking about laboratory samples traveling from one part of the world to another, there's going to be logistical challenges that are going to be involved. So that is going to be important to consider. I do believe there is a sequencing, some sort of sequencing organization that has a very good sequencing platform in Nigeria. So there may be options that you can explore just locally if you're from Nigeria. I'm not sure. Just saying, right? But closer options are available, I believe. Right? When you're talking about sequencing, when you talk with them and you engage them, I think it'll be good to explain the type of sequencing project that you are interested in doing. So for us, our specialization primarily is in animal health related stuff, specifically for viruses or for bacteria. So if your sequencing project is going to be on plants or on a different type of organism, right? Then it is actually probably better to find a specialist who is more fit for that particular project. And I think the value that you'll get from that project will be much more than just any random sequencing organization, if that makes sense to you. Yeah. So I hope that answers a little bit.
Yeah, thank you very much, Isaiah. There is actually a molecular diagnostic lab in Nigeria. Well, the only thing we're trying to achieve is that we want to be able to expand what is currently available in Nigeria, because yes, there is the PCR, the sequencing, the next generation sequencing, everything is available, and you can actually contact me if you are interested in doing that for your project. I can link you up to the laboratory so that you can do your activities and complete your project easily. But all we're trying to do is to expand that opportunity, and that's the reason why we are currently doing this. So hopefully, if you can get that, just contact me through the email address that I've sent, then we can sort things out within the country. Yeah, so thank you very much. Oh, okay, so there is another question. Can we have opportunity of knowing how to analyze the results of the PCR? Yeah, Josiah. Yeah, that's in the chat box, not in the question and answer.
Okay, sorry, I was trying to, too many tabs opening. Okay, thank you. Question is, can we have the opportunity of knowing how to analyze the results of the PCR? Yes, I think so. I tried not to focus too much on the mechanisms of PCR today for this presentation. I think there's the technical aspect of PCR will require its own presentation, and you can actually find a lot of great tutorials, resources, videos online, just on YouTube itself, about PCR and how it works, and how the signal is artificially boosted, right? But in general, I think a good place to start is, and how I myself learned it is like with conventional PCR. It's just a visual signal, and I think a bigger part, and not just the signal itself, but designing your assay is going to be a big part of the PCR as well. I'm not sure if my colleagues in the next session will maybe go through a little bit. Maybe we can put something there. But yes, I think the good place to start if you're looking about or thinking about how to analyze PCR is just go on YouTube, type in PCR resources or tutorials, and there will be plenty of great resources out there for you. So thank you.
Yeah, thank you very much, Isaiah. So yeah, there are actually different ways in which you can do that. So, and it depends on the type of PCR we're talking about. So yeah, I think the best thing is just to know what you're dealing with and then go to the YouTube to get some information from there. But believe me, it's not a very big issue. Molecular makes things easy. So yeah, it's what you can actually access and understand by going through it. So just know what your assay is all about, what principle you're working with, and what type of PCR, and then you can go online and get some information from there. So it's not a big issue. Thank you. I don't know if there are that satisfies your curiosity. So we have another question, and we don't have time. Josiah, please, can you quickly go through and give question and answer?
Yeah, in the questions and answers, yeah. Kadar Ali Said, your team in Malaysia, are they working together with Pan-African vaccination quality control to strengthen each other? Thank you so much for the question, Kadar. No, we are not. I'm not familiar with the Pan-African Vaccination Unit or that organization. All I can say is that at the present, we are interested in the African continent, and we are working with different local partners, EVMEDS being one of them in Nigeria, to just talk about molecular diagnostics. So we don't do any work related to vaccination of animals. The only related work we do with vaccination is with one of the MNCs here in Asia, where we are doing some vaccine efficacy testing for their products. So that is part of the work that our lab using molecular technologies is doing. But we are not actively going out and vaccinating animals or working with organizations that do that necessarily. So I hope that answers your question, Kader.
Yeah, thank you very much. So Khalid, if you actually have a particular request in mind and you would like us to find a mutual brand, I think we can take the discussion further from here. You can reach out to us through email, and then let's see how we can work together if that can help what you're doing in any way. We about Africa, and we desire to actually bring some forms of, let me say, ease into how things are being done. So if you think with the Pan-African vaccine thing, you see a gap that we may be able to help you fill, yeah, we are open to that, and then we can see how we can work that together. So just reach out to us through the email, and then we'll get the discussion going. Yeah, thank you very much. So let me see. All right, I'm still going to drop my email in the chat box for those that desire to get. I've done that before, but I'm going to do that again right away so that you can have access to it, and then we can keep the conversation going. All right, I'm not sure there is any other question, so we may want to draw the curtain now that we are 5 minutes even behind the schedule. So we're going to see you all next week, because like we mentioned, we're trying to go step by step and make it more practical. For the person that asked regarding the, I think mentioning the phylogenetic tree, I wouldn't want to miss that. Yeah, so phylogenetic tree is another level of molecular. So after you finish your PCR, you've done your sequencing, and then you will now start with bioinformatics, and then we can start talking about phylogenetic tree. So yeah, we can do that, but that is another level entirely. But gradually, like I said, we just want to be systematic about our approach. So come next week, learn more about molecular, and then next week, and then next week, and then you should be able to get there one of these days. All right, thank you everyone for participating in this webinar. It's been really great and interesting. We promise to make the recording available and the materials available to you so that you can go through probably before next week to have a kind of fundamental understanding of this because it's a building block. You need this like a foundation to be able to lay what you're going to be learning next week. And then we promise to make it as interactive and practical as possible so that we can have broad learning and then take something very crucial away from this series of webinars. Once again, thank you, Josiah. Thank you, LIR team, for coming around for this presentation, and for everyone that has come to participate. We are very, very grateful for coming around, and we will see you next week. Tell your friends and colleagues what they have missed today so that they can join us and learn one or two things. Thank you, everyone.
Thank you, Dr.
For making this possibility, and have a wonderful time at work or wherever, and enjoy the rest of the way. Yeah. Bye for now.
Thank you, everyone. Have a great Wednesday. Take care.
Curry. Curry.